We'd like to get started. I'm going to, as usual, I'm going to introduce the introducer of the speaker who's actually going to talk. And our speaker today is Hedy, who's going to be talking about regulation of craving from neural mechanisms to treatment development. And Hedy's work is really exciting, and I think you're going to really enjoy it. But I also wanted to mention that next week we're having a special Grand Rounds in memory of Leonard Hill. I'm not sure everybody here knows him. Leonard, in my view, is in the pantheon of historical leaders at Yale New Haven Hospital Psychiatry. You think of Tom Detry, who transformed the nature of an inpatient psychiatry, essentially creating the framework that most inpatient psychiatry units at Yale and most other institutions follow. Malcolm Bowers, a transformative leader, Craig Nelson, and Leonard. Leonard and Ralph Hoffman essentially created the model for our intensive outpatient psychiatry programs at Yale New Haven Hospital. And for several decades, Leonard was the main kind of clinical director of the Yale New Haven Intensive Outpatient Clinic, or day hospital, if you will. A wonderful leader, compassionate and effective therapist. And so we're really thrilled to have a special day to remember his contributions. And the Grand Round speaker on that day is going to be Fromo Walsh from the University of Chicago, who's going to be talking about strengthening family resilience, which obviously is an important topic. The following week, the 11th of November, you have the day off. It's Veterans Day, and there's no Grand Rounds. And then on the 18th, so 3 weeks from now, the speaker will be Carrie Bearden, who's going to be talking about psychosis risk mechanisms. She studies people from a developmental and genetic perspective, and she should give a great talk. So, and after that's Thanksgiving. So, some great presentations, a day off for Veterans Day, and with that, I will introduce Stephanie O'Malley, who will introduce our speaker. Thank you.
Before we get started, I have an announcement to make as well. So we are really trying very hard in the Grand Rounds Committee to make sure that people have the opportunity to go to the lunch after a speaker if they're particularly interested in that topic. We like to focus on inviting trainees and junior faculty. Pretty soon you're going to get an email with a list of all the speakers for this year and who's considered sort of the host for that person. And if you have some interest in going to the lunch or perhaps meeting with the speaker if they're here longer than their talk, I'm going to ask you to please email the host of that event and indicate your interest. You'll also copy Tricia Dahl, who coordinates for us, but that way we can try to make sure that you have a chance to come. Now, obviously, everyone won't get to go to everything they want to do, and we're going to try to look to see that everyone has an opportunity to attend one of the lunches. So I just wanted to announce that for you to keep a lookout for that in your email. The other thing is here we go. Good morning, welcome. This Grand Rounds is accredited for CME by the Yale School of Medicine. If you wish to receive credit for your participation, please be sure to sign the attendance sheet by the refreshments. At the present, I would like to acknowledge on behalf of the department that there are no commercial sponsors of this Grand Rounds and that I've reviewed today's lecture and faculty disclosure and found no conflicts of interest. Okay, so with that, I'll move on to a very brief introduction of our speaker, Hedy. I think she probably doesn't need an introduction to our group, but I think it's important to kind of get a little bit of her background. She received her bachelor's degree from Columbia in psychology, and she also pursued her PhD at Columbia as well in the Cognitive and Affective Neuroscience program and received a degree and a PhD in psychology. We had the pleasure of having her join our faculty in 2010 as an assistant professor. But Hedy is not just part of our department, but she holds appointments in several other departments and programs at Yale, including the Cognitive Science program, the Interdisciplinary Neuroscience program, the Department of Psychology, and the Yale Law School. She has already received numerous awards, including Early Career and Travel Awards from NIDA, the Committee for Problems for the CPDD, as well as other fellowship and travel awards. And at Yale, she's had a very active program of teaching, which is unique, and has runs a course over in the Department of Psychology and has done some teaching at the law school too. And she has an active research program. The overarching goal of her research is to understand the psychological and neurobiological mechanisms that underlie addictive disorders, with the specific aim of developing and improving treatment. Her primary focus has been on craving and the regulation of craving as they relate to drug use, treatment, and relapse, and we're going to hear about this today. It should be a very exciting and interesting talk. So with this, I'm going to turn the floor over to Dr. Kober, and if you join me in welcoming her to the podium.
Thank you for the very kind introduction. It's a pleasure to be here. It's a pleasure to give Grand Rounds. I actually gave Grand Rounds here 4 years ago, and as I was preparing the lecture for today, it was really incredible for me to see how much work we've gotten done since then. And I'm gonna start my talk by kind of holding a moment of gratitude for a few people without whom this talk wouldn't happen today. And the first person to whom I'm gonna really express a debt of gratitude is Bruce Ransville. I literally would not be standing here if it wasn't for him. I came to Yale because of him, and he's a dear mentor, and I still miss him. I also owe a very large debt of gratitude to Mark Potenza and to Kathy Carroll, who really continued where Bruce left off and held me as mentors since then and until now. And also, I don't want to forget their wonderful group of trainees, Iris, Elise, Patrick, Jinsong, and Sarah. I counted last night, we have about 20 manuscripts together from the last few years. And although I'm not going to talk about a lot of that work today, it's been a real pleasure. Very, very important is also to all of the very, very wonderful people in my lab. They're truly a dream team. I want to specifically highlight Ross McLean, Becca Boswell, my graduate student, Maggie May Mel, Matthew Schaefer, and Shosuke Suzuki. I'm highlighting them specifically because today I'm going to talk about data that they produced and spearheaded specifically, but of course all these other people contributed to some of the data that I'm going to talk about today. I'm also going to highlight our website down here. You'll see as the talk goes on that a few of the references are marked in orange. Those are references for papers specifically from our lab. There are obviously going to be references that are from other labs as well. Anything that's in orange is available for download on our website if you're curious. I also, of course, want to thank my very many and wonderful collaborators, both at Yale, including Stephanie, John, and Alan at the CTNA, Rajita at the Stress Center, and Godfrey and Michael Stevens up at the Institute of Living. I'm going to show today some of the data that we collected together, and of course, my collaborators and former mentors at Columbia, University of Colorado, my former student who's now at Dartmouth, and then some collaborators at Carnegie Mellon. I'm going to show today some data that we collected together as well. Last but not least, much gratitude to all the funding sources. I would be nowhere if it were not for the National Institute of Health, especially the National Institute of Drug Abuse. These are various grants to various people, including me, who've supported the work that I'm going to show today. Okay, so I'm going to start actually with a kind of a broad comment about addiction in general, but I'm going to really jump very quickly into the basic processes really of cue reactivity and craving, which are of course central to addiction. I'm going to specifically argue, despite this being a controversial topic, that craving predicts eating and drug use and relapse, and I'm going to use meta-analysis of outcome data to make that argument. I'm going to then talk about the neural mechanisms of craving using meta-analysis of neuroimaging data to describe describe the regions that are most consistently associated with that process. I'm going to then talk about the regulation of craving, the ability to use strategies to reduce the experience of craving, and specifically the ROC task that we developed for that purpose. And I'm going to show you data from cigarettes, food, cocaine, methamphetamine, and alcohol. We've really collected a lot of data on this over the last few years. I'm going to make some specific, both theoretical and data-driven arguments regarding the neural mechanisms that might underlie cognitively based or CBT-based regulation of craving versus mindfulness. I'm going to talk about how we might use this information to develop trainings in regulation of craving, and then going to end on conclusion and future directions. And since we're actually already 10 minutes into my allotted time slot, I might skim some parts in the middle. We shall see. So my first general comment about addiction, obviously defined as a persistent and maladaptive use of psychoactive drugs, is that you might not know this, but it's actually the most prevalent of all psychiatric disorders if you really look at it as a group of disorders. Most people think that mood disorders are more prevalent. Many people think that anxiety disorders are more prevalent. But in fact, with a 35% lifetime prevalence across all of the addictions, they represent the most common, afflicting 2.5 billion individuals worldwide. That's really, really a lot. And what makes it even more of a compounded issue is that it's a chronic relapsing condition. So individuals who qualify for that diagnosis at one time are likely to qualify for this diagnosis at another time. And it's really personally devastating in a few different ways that sometimes at least depend on the kind of drug. I'm going to just show you this very, very quick clip from a woman who suffers from a stimulant use disorder. Oh.
It's taking my children, my freedom, my teeth, my looks, my ability to function in society.
So it's taken my children, my freedom, my teeth, my looks, my ability to function in society. This might not be true, of course, for all kinds of addictions. Other kinds of addictions are associated with different kinds of personal costs. For example, cigarettes kill over 480,000 people per year. This is just cigarettes alone. But also, there's a staggering social cost associated with addictions. In the U.S. alone, $700 billion annually are being spent on things like healthcare, law enforcement, loss of productivity in the home and the workplace, and premature death and disability. And of course, that number is so large, I'm sure that none of you can really wrap your heads around how much money that is. And so just to compare, some of you know I was born and raised in Israel. The GDP, the gross domestic product of Israel, is $242 billion. So the U.S. spends almost 3 times as much covering the costs of addictions. But to kind of come into thinking about one of the basic processes in addiction of cue activity and craving, I have to now take you 100 years back to Ivan Pavlov. I'm sure you guys have all heard about Pavlov and that his name rings a bell. Back in 1901, he discovered in a series of experiments that when he presented his dogs with food, if he repeatedly paired it with a bell, they began to salivate even before the food was served to the ring of the bell. And at the time, he called this psychic secretions. Luckily, we don't call that psychic secretions anymore. But he did come up with this finding that ended up serving as the basis to most learning-based models of behavior that we use today. And the basic finding goes something like this. Food is an unconditioned stimulus. Food reactivity is an unconditioned response, or a set of unconditioned responses. And therefore, cues that are repeatedly paired with food become conditioned stimuli. So they actually begin to be associated with the conditioned responses, which we have come to call cue reactivity. Again, this depends on kind of repeated pairing of these cues with the unconditioned stimulus. And so back in the present, there's maybe in the last 20 or 30 years, a lot of research that has shown repeatedly that cues that are paired with food, and that might include sometimes even the people that you eat food with, certainly the place that you eat food, pictures of food, so the sight of food, come to elicit cue reactivity, and also the experience of cue-induced craving. Now, people have long thought that the physiological experiences associated with cue activity, and for food that might include salivating, but also ghrelin secretion, digestive juices, having salivation in your mouth, all of these are consciously experienced as craving, so we can really think of cue-induced craving as either a type of cue reactivity or an emergent property of cue reactivity. And just in case the explanation itself didn't make sense, I'm going to start with a little demonstration. It's morning. Hopefully most of you haven't had a full breakfast. And so I'm going to ask you to look at this really beautiful omelet. Maybe you can check out these like little strings of melted cheese and imagine that it's warm and you're about to take a bite. You can also look at this bagel. There's some fresh avocado on it and some lox. Maybe some of you really prefer sweet food a little bit more. These are obviously chocolate croissants. I really love the chocolate glaze on top. This is one of the most craved photos in our database. People typically kind of go down with their eyes, right? Like right where the glazing of the maple goes. And maybe if you just want to go all out for sweets. This is a chocolate brownie. I think there's some salt and almonds on top, and of course, this drizzle of caramel. And as you're looking at these images, hopefully you can really get a feel for them. And then I'm going to ask you, did any of you salivate to any of these images even a little bit? If you want to give an answer of more than 2, please raise your hand. All right. So you just experienced cue reactivity. And did you feel like you wanted any of the foods that I just presented? If you were going to give it more than a 1, please raise your hand. So you also just experienced cue-induced craving. Thank you very much. And so hopefully this gives you a good kind of sense for what this feels, and also an idea, given how many people really raise their hand, this is an exceptionally common phenomenon. And of course, since I'm going to talk a lot about addiction, I'm going to kind of repeat what may be obvious for some of you. Drugs are also unconditioned stimuli. Drug effects are unconditioned responses. And therefore, cues that are repeatedly paired with drugs become conditioned stimuli that lead to a set of conditioned responses that are typically related to drug effects themselves. And so drug cues come to give rise to drug cue reactivity and to drug cue-induced craving. And what's really interesting is that there's so much really data showing specifically that drug cues give rise to cue-induced craving that the first meta-analysis on this topic appeared in 1991, right? So this is really a very fairly well-established phenomenon in itself. But if we come to agree on what craving might be defined as before we go on, then we have to start from the fact that it's a really well-established neurobiological process that we as humans define and experience as strong desire or wanting. But what's really important, I think, is that this kind of conscious desire and wanting has been discussed by Eastern and Western philosophers for literally centuries. The Buddha 2,500 years ago said that craving was a source of all suffering. Baruch Spinoza in the 1600s said that it's the very essence of man. Bertrand Russell says that it prompts all human activity. And while I don't yet have data that it prompts all human activity, there are data to suggest that this is an exceptionally common experience. Most surveys show somewhere between 94% to 100% of adults reporting feeling craving on a semi-regular basis. When we come to think about it from a definitional perspective, we typically think about it as a multidimensional construct that has this physiological component of cue reactivity, an affective component or the feeling of wanting, a cognitive component, which is both the thought, I want this, but also expectancies. If I have this, I'll feel better. If I don't have this, I'll feel worse. And then a motivational component, right? Really important, that's associated with the impetus to act on these thoughts and feelings. And it's exactly this cluster of definitions for craving that has been considered a key process in addiction for quite a long time. But really only in the last 3 years, it has been added as a diagnostic criterion for the cluster of substance use and addictive disorders. And it was added as a diagnostic criterion based on a good amount of clinical data linking craving to drug use directly. People often report that craving is their reason for relapse. There are many studies that have shown correlations between craving and drug use, both prospectively, retrospectively, and using ecological momentary assessment studies. And also there's some number of studies that have shown direct prediction of craving on drug use and relapse across various substances and across different time resolutions. So people who report more craving today are more likely to use drugs later today, but also more likely to relapse a week from now, a month from now, et cetera. But despite this clinical data, and despite the fact that the clinical data were strong enough to kind of upgrade craving to a diagnostic criterion, there's really an important caveat, which is the role of craving in drug taking really remains controversial. And people have argued for this in a few different ways. So people have argued, for example, that cue-induced craving is actually less important than tonic or questionnaire-based craving, and that those two are actually more dissociable from each other than some other people think. Some people have argued that it's really only relevant during cessation or during abstinence, that people who are actively using, for them craving is actually less than a predictive factor. And especially animal researchers have argued for the presence of unconscious habits or unconscious cue reactivity that drives behavior in the absence of the conscious experience of craving. And because I'm really interested in this, and what I'm really interested in is really in the causal role that the conscious experience might have, I really wanted to know what we might resolve this. And one solution for this is to actually conduct a meta-analysis across all of the studies that have been published. Now, meta-analyses in general, in statistics at least, are really considered a way to combine the results of multiple studies into a single aggregate statistic. And so you look at a bunch of studies that describe a particular research area and aggregate their statistic into a single one that might describe the strength of the evidence. You can think about it as a study of studies. Now, importantly, this is the strength of evidence and the consistency of evidence is one of the first requirements when you are about to determine causality in biological processes. There's a really nice paper that delineates this kind of logic, and that makes meta-analysis an ideal tool for this particular dilemma. I also want to put in a plug that it's also an ideal tool in general for the replication crisis that's currently plaguing psychology and just beginning, I think, to plague our field as well. And so the kinds of study that we're interested in here are also ones that have a particular temporal structure. So at time one, there's either cue exposure, cue activity that's being measured, cue-induced craving that's measured, and craving questionnaires so that we can really separate the effect of cue exposure, cue activity from various kinds of craving. And then at time two, that can be either minutes or hours or days or months or years later, but the important part is that it's later, there's some kind of outcome measured. And I'm actually going to show you two meta-analyses that I've been working on with my wonderful graduate student, Becca Boswell. The first is specifically looking at the relationship between these factors and eating or weight gain. This paper is already published. And then one that we're actively working on right now with some of the other students in the lab, including Darby, who I see here, specifically looks at the relationship between craving and cue activity and drug use and relapse. And the idea that what we're specifically looking for is not only this temporal relationship between the two measurements, but statistics that relate those two measurements between time one and time two. And for all of our meta-analyses, and I'm actually going to show you a few today, we use the preferred reporting items for systematic reviews and meta-analysis, or PRISMA. And I'm going to show you, I'm going to go through this, I'm going to take maybe a minute or two to go through this because I really want you to understand how much work and how much data really goes into producing the kind of results that I'm going to show you. So this is for the food meta-analysis on the left and the ongoing meta-analysis that we're not done yet with for drugs on the right. And so first there's the identification stage. We typically go to different kinds of indexing databases and search for any paper that has keywords that are relevant to our search. So for example, for food, we might look for food, cue, craving, cue-induced craving, anything like that, weight, eating. For drugs, we might look for each drug separately along with cues, craving, and the like. And so we really pull initially thousands and thousands of records off of these databases that we examine initially for inclusion. And we also do additional record searches either through looking through reference sections or doing reverse searches on papers that we've already included. And again, you can see this culminates in thousands of papers being examined. We then discard duplicates. Typically, there are many, many duplicates, so you have to keep pretty close records. And then we go through a stage of screening where, again, we screen thousands of papers to exclude papers that, for example, might not include humans, might not actually be measuring craving even though they mention craving, might not actually have an outcome, et cetera. Then we assess for eligibility, which means that we actually look at the full text of each article and determine whether all of the inclusion criteria are satisfied. We might exclude papers for not actually having statistics, which, by the way, you'd be shocked how many papers actually have all the data but don't actually report statistics. It's really sad. It's not prospective sometimes, or actually don't have the measurements even though they administered the right tasks. And then eventually we come up with how many studies to include. So in this published meta-analysis, we include 45 45 studies with 69 statistics, meaning more than one statistic per study, representing 3,292 participants, right? So this is bigger than any single study that probably any of us have ever run. In the drug meta-analysis, although we're still maybe halfway through, you can see there are many, many more studies to be included, 94 so far, 298 statistics, representing 16,302 participants. And if you're curious, I think we're done with all the alcohol studies. We're still going through cigarette studies, and I think that's about the number of cannabis studies that we're going to be able to include, but that's what we have so far. So first, looking at the relationship between cues and craving, eating, and weight gain. So the first thing that we looked at is really what is the effect of cue exposure on eating and weight gain. And we discovered a perspective R, so aggregate across all of the studies that just showed cues and then measured the effect on behavior of 0.29. This is a medium effect size, which is quite significant. We looked at cue reactivity, meaning looking at the magnitude of cue reactivity responses, so how much salivation, how much heart rate increases, and linking them to the magnitude of eating and weight gain, and found a medium to large effect size of 0.35. For tonic craving or questionnaire craving, the effect size was again medium for 0.3. And for cue-induced craving, again, a medium to large effect size at 0.35. Now, this is quite strong evidence, and remember, this is all perspective, that cue-induced craving specifically predicts eating and weight gain over time. And I also specifically want to say that these are both short and long-term outcomes. Now, the combined effect across all of the studies and all of the statistics in the database representing 3,292 participants is again a medium effect size, a combined effect, suggesting that all of these processes together account for about 11% of the variance in eating and weight gain, which is, I think, more than we know of any other factor influencing eating and weight gain directly. Now, just to say ahead of time, since most of the data that I'm going to show you today, the experimental data at least, is going to deal with images of food and drugs, when we specifically looked at images as cues compared to real food, they both were associated with a medium to large effect size, suggesting that specifically images as cues have a strong effect on eating and weight gain. Also, when we looked at outcomes related to eating immediately in the lab versus weight measured up to 2 years, we actually see somewhat better prediction at the long-term outcomes, which I think is really interesting and really important. Now, some of you might look at this and say, well, you know, a lot of people measure these things and they have null results and they don't publish it. What do you do about publication bias? Well, there are actually analyses that we can run to formalize against publication bias. And what these analyses come up with is essentially a number of studies that would need to be published with a null result or the opposite result for this meta-analytic statistic to become null itself. And the fail-safe N here is 4,186 studies that would need to be published for this result to no longer hold. I highly doubt that there are that many studies published with the opposite or unpublished with the opposite result. Now, again, the drug meta-analysis is still ongoing, but when we look at the relationship between cues and craving, drug use, and relapse, a very similar picture is emerging. So for cue exposure, we're finding a medium effect size of 0.26, for cue activity 0.32, for tonic craving a little bit lower at 0.24, and for cue-induced craving, a medium effect size of 0.31, again, suggesting that cue-induced craving specifically is a very, very important factor in here. And here I'm also going to show you a particular analysis that's, again, important to the kind of data that I'm going to show you today. When cue-induced craving was measured with a single item question, it showed across all of the studies in the meta-analysis a 0.28, a medium effect size, predicting drug use and relapse. So it's really very, very powerful prediction, even with just asking the single question, how much craving do you feel for this? The combined effect across all of the studies in the meta-analysis so far is a 0.26, with 0.24 for immediate outcomes. And again, with a stronger effect of prediction for longer-term outcomes. This is aggregate over all the outcomes in the meta-analysis are 0.3. Now, maybe one of the most astonishing numbers is the fail-safe N for this meta-analysis. Remember, we're only halfway through. We would need 323,000 studies to make this effect null. That would be unpublished with either null results or the opposite results, suggesting again that this is very, very strong. And remember, this represents 16,302 participants. So I think that we can begin to make some comments about whether craving actually has a causal effect on behavior. But for now, I hope that you can at least agree with me that cue-induced craving is an important contributory factor in eating, weight, substance use, and relapse. So far, so good. Anybody has questions? All right. So hopefully I've convinced you now that craving predicts eating and drug use, at least to some degree. And now we're going to talk about the neural mechanisms of craving. Now there's obviously a rich animal literature on this, using kind of conditioned drug use as a basic process that has implicated the involvement of dopamine in various brain regions, including both the ventral and the dorsal striatum. But the emerging human literature over the last 2 decades now has really been inconsistent, with people arguing quite vehemently over the role of either the ventral and dorsal striatum, is insula involved or not, is the amygdala involved or not. There's been quite a lively debate. The first time that we looked at the kind of the literature itself was in 2011, when we first had the idea to do this, do a meta-analysis, and we found about 50 neuroimaging studies that have looked at this. But now that we've waited through 2016, another 68 studies have been added to the literature, so you can see that the rate of studies is really increasing quite quickly. And still, the regions that have been reported vary dramatically across the different drugs, the different paradigms, the different cues that have been used, and maybe even the different analysis procedures across everybody. And so here, again, we're using a meta-analysis of neuroimaging studies. Luckily, I've done quite a lot of work on these kinds of methods, doing meta-analyses on emotion and other kinds of content areas. But here, we were specifically looking for brain imaging studies with adult participants and drug-related stimuli. And the adult participants were also drug-using participants in this case. I'm not going to go through the PRISMA in quite as much detail, but I'm going to show you that there's the same kind of stages apply to this. We're not yet writing up the paper, but we finished all of the analyses. And we came up with 118 studies, including 168 contrasts and representing 3,513 participants in these kinds of studies. And this is work that I mostly worked on with Ross McLean, postdoctoral fellow in my lab and in the VA. And in case you're curious, the breakdown with drugs is mostly cigarettes, almost an equal number of alcohol studies, many fewer for cocaine, for opioids, for gambling, for marijuana, and for methamphetamine. You'll also might not be surprised that most of the studies used images, although many used videos, audio, and some used also a mix of stimuli, and then most of them are also fMRI studies. We can, of course, analyze for each of these factors if we're interested in its effect. I'm actually you're gonna have to trust me that the methods are really careful and have been well established. We both published with this method specific meta-analyses and also published some, this is a method paper on kind of different methods of meta-analysis. And so I'm just gonna skip to the results. And what I'm gonna show you are regions that are most consistently and specifically activated across different kinds of analyses. So the first analysis across all the studies and across all the drugs, these would be the regions that are most consistently activated across all of the Q presentations, all of the drug cue presentations. And what you see here, this is a sagittal slice, right? So for those of you who don't know, this is if, let's say, a crazy axe murderer chopped my brain in half and this side of my brain fell off and you're just looking at it this way. The middle is a coronal slice, that's you took my face off and it fell forward and you're looking at it this way. And then an axial slice, it's like you took the top of my head off like a top of a pot or something. And so what you're seeing here is the ventromedial prefrontal cortex, both the ventral and the dorsal striatum and the subgenual cingulate, which is really important for a few different kinds of arguments that people have made. Of course, we can look, for example, at further analyses at people who are heavier user and lighter users across the database to see whether there really is a separation between ventral and dorsal striatum. We see here evidence for the posterior cingulate cortex, the thalamus, and of course the amygdala bilaterally. And hey, lo and behold, the insula with that many studies actually does come up across all of the drugs. This is something that surprised even us because across the other meta-analyses that have been published now 4 or 5 years ago with just a few studies, they didn't actually find evidence for the insula, which other people have argued quite vehemently for. And even we didn't find it when we did interim analysis with about 70 studies. So this is really exciting. What I think is also interesting is that when you look at each set of studies for each drug separately, there's actually quite some variability. I'm not going to belabor what this means yet because we're still kind of pondering over the meaning of this. But when you look at cigarettes separately, alcohol separately, and stimulants separately, you can see that there's actually quite a bit of variability amongst the different drug groups. And we may, with some sophisticated multivariate analyses, be able to start to parse what these differences might be across all the drugs. One of the analyses that I was, of course, most interested in is looking at only those coordinates that were explicitly reported as correlated with a conscious experience of craving. And one might imagine that if cue-induced craving is a type of cue reactivity, and these really represent cue reactivity, that we'd really see a subset of these regions activated for the conscious experience of craving, and that's exactly what we're seeing. And that's this region of ventral medial prefrontal cortex and ACC, this region, which is ventral striatum and subgenual cingulate, this tiny, tiny activation in the amygdala as well. And so we really are starting to kind of inch towards, and of course we have more analyses to run, what might be the regions that are really most specifically associated with a conscious experience of craving. And we also have a grant now under review, hopefully it will be reviewed next week, to try to do kind of use machine learning methods to develop a multivariate predictor of the craving experience itself and actually test it on some of the data that we're collecting from drug users. And so what I will say for now is if we have to understand what this network of region means, we can say that it's consistently activated during cue activity in the experience of craving, that there are some between drug differences that may be meaningful, that it's certainly along the dopamine pathway, which is not surprising since we talked about craving as a cue conditioned response. But people also look at this often and say, oh, it looks like there's default mode network activity, which might be related to the fact that these are very self-relevant cues. But one of the things that really struck me is that these are regions that are commonly activated in emotion. This is a slice from one of our emotion meta-analyses. This is from 2008. And you can see that there's actually quite a bit of overlap. And in another analysis, here you can see that it's also particularly overlapping with both positive and negative emotion. And so one analysis that we could do here to try to parse this out is to try to, let's say, separate individuals who were scanned while they were abstinent versus not, right? You might imagine that there'll be some better differentiation between more positive or more negative emotion. But what's maybe the most important is that these regions would be the target for modulation if we were going to image the regulation of craving, which is, luckily, what I'm going to talk about next. So I talked about craving being a diagnostic criteria for addiction, but there's, of course, many, many other diagnostic criteria, including using larger amounts and for longer than intended, continuing use despite knowledge of ill consequences, and wanting to stop but failing, all of which really represent a failure to regulate craving and drug use. And this is really consistent with addiction being formulated across many, many different models of addiction as a disorder of regulatory failure. And indeed, poor regulation is considered both a risk factor and a distal causal factor for addiction, a proximal causal factor for particular instances of drug use, and also a maintaining factor for the disorder itself. And this is consistent with data suggesting that addictions are very difficult to treat, and indeed, even with the best of our treatments, 30% of individuals drop out before treatment completion, about 30% of individuals achieve abstinence, and then there's very, very high relapse rates across disorders. On the other hand, there's of course also the insight or the data that active regulation of craving decreases use. And some of this comes from treatment studies of cognitive and relapse prevention treatments that are really the most effective treatments that we have for reducing substance use. And they specifically have modules that focus on identifying and avoiding drug cues in high-risk situations where drug cues might arise and craving might arise. They teach strategies for regulation of craving. And there's at least some data that suggests that acquisition of coping strategies, including strategies for the regulation of craving, are associated with reduced relapse. And some of this work was done by Brian Killick, who's sitting right here. I really love that line of work. And there's also data from ecological momentary assessment studies that have done really careful perspective work showing that momentary application of cognitive strategies in moments of craving in people's day-to-day lives is associated with reduced craving and reduced relapse. And that was done both with ecological momentary assessment studies and in perspective studies. And people have indeed hypothesized that the ability to regulate craving and use these kinds of strategies is one of the mechanisms of action of CBT in general and potentially of most treatments for substance use, which suggests that regulation of craving is really a key component in promoting abstinence. And so to test this directly and mechanistically, we developed the regulation of craving task. This is actually the work of my dissertation, which I'm going to talk about just briefly. And it really was meant to test the causal effect of strategies on the experience of craving and to be able to trace the neural mechanisms that underlie both craving and the ability to regulate it. This is a trial course a trial time course in this task. Looks a lot like kind of classic emotion regulation paradigms mixed with maybe delay of gratification fixation, participants look at a fixation cross and they receive a particular instruction to either feel craving or to regulate. They see an appetitive stimulus. This can be across the different studies. I'm going to show you food, crystal meth. Hopefully none of you are reacting to this at all, cocaine, and cigarettes, and another fixation cross, and then they're asked trial by trial to report the strength of their craving. In the first two studies, we did this with cigarette smokers. We showed them both food and cigarettes, and we gave them two kinds of instructions, so really ways to think about the cue that's about to appear, and they're trained on how to follow these instructions before ever beginning either the behavioral session or the fMRI session. The first instruction is based on the natural response to the cues in some of our pilot work, which is really focusing on the immediate feeling associated with eating, smoking. When we ask, for example, cigarette smokers what happens to them spontaneously when they look at cigarette cues, they almost always reported that they immediately think about how good it would be to smoke it. And so just to keep good controls, we tell them to do exactly that. If you're shown a picture of a cigarette, you should think about how smoking will make you feel when you light up, focus on the smell, and how smoking the cigarette will taste. And the cue word we used for this condition was the word now. The second instruction really came straight out of the CBT manual for substance use disorders and oriented participants to think about the negative consequences associated with smoking or eating. So if you were shown a picture of a cigarette, you should think about the consequences smoking will have for your lungs, for your heart, and for your likelihood of developing cancer. And the cue word we used for this condition was the word later. And so again, if you were a participant in my study, you'd see a fixation cross, you'd get a strategy cue, either the word later or the word now. You'd see a picture of a cigarette or a picture of food if it was in that first and second study in the series. And then you'd see another fixation cross and you'd be asked to rate your craving. So for example, if I saw the word now and I saw this picture of a brownie, I might think about how the brownie is really hot and the ice cream is cold and I really love cherries and the chocolate sauce. And by the time that I think all of that, I definitely would be salivating. And then I'd be asked to rate my craving. On the other hand, if I saw the word later, and let's say I saw this picture of a cigarette, I would think about tar in my lungs and the risk of developing cancer and how bad it is for me, and that it's a really bad idea for me to smoke. It makes it hard for me to run, and then by the time that I think that, I'd probably be asked to rate my craving. Now, now this might not seem so shocking in retrospect, but when we first published this, we actually got a lot of pushback. We went through, I think, 4 rounds of reviews at drug and alcohol dependence before the reviewers actually trusted that these data were real, because they thought that it's not possible, but we've now replicated it many times. You can see high craving when participants see pictures of food and follow the instruction now, and significant reduction in craving when they think about the negative consequences of eating. And we see the same pattern with cigarettes when they report high craving, when they think about the immediate consequences of smoking, and significantly reduced craving when they think about the negative consequences. Now, both my group and other people have replicated these data now with cigarette smokers. This is actually an EEG study from a group in the Netherlands. This is with stimulants. This is my former student Rich Lopez in a paper with meth users, and Matt Schaefer is currently working on a paper with cocaine users. And Nazar Naqvi, my colleague at Columbia, has done the first behavioral study on alcohol. We're working now with the pilot data from the CTNA and Shosuke Suzuki on a paper with alcohol. And then with food, there's actually been a lot of papers that have followed up using our task that have shown consistently that people, even individuals who are obese, can reduce their craving, although what's really emerging as really important is individual differences in the ability to do this. I'm going to talk about this more in a minute in the context of the imaging data. So in the first imaging study on this, we compared brain regions that were more active in the now condition compared to the later condition. This is the craving condition. And what we see is this network of regions that's very, very consistent with the meta-analytic findings. So increased activity in these predicted craving regions, consistent with the behavioral report that they were feeling more craving in the ventral medial prefrontal cortex, in the ventral tegmental area, subgenual ACC, and ventral striatum. Now, when we reverse the contrast and look at regions that are more strongly recruited in the regulation condition compared to now, what we see, first of all, is activation in regions that were previously associated with cognitive control, including dorsolateral, dorsomedial, and ventrolateral prefrontal cortex. And then we see relative deactivation in all of these craving conditions here. Which I think is quite remarkable, including the ventral medial prefrontal cortex, the subgenual ACC, and the ventral striatum. Now, one of the important points here is that it suggests that under these laboratory conditions, when we bring these cigarette smokers in, they're not generally impaired. It's not that they're unable to regulate their craving. They might not be doing it in everyday life because they are smoking, but when we tell them to do it in controlled settings, they're able to do it, and they're able to recruit the brain regions that are necessary to perform this activity. And if we focus specifically on this activity in dorsolateral prefrontal cortex, we see a nearby region. This is a whole brain analysis that shows really this effect of individual differences. Those individuals who recruit the DLPFC most strongly are the ones who report the lowest craving on the task, suggesting that this region that really was previously associated with cognitive control tracks negatively with craving. So if you recruit it more, you actually regulate better. And we see the opposite pattern in the ventral striatum. So the more activity in the ventral striatum, the more craving people experienced. And so what we see here is a pattern where the ventral striatum tracked positively with craving and the DLPFC tracks negatively with craving. And this suggests a few potential relationships between these regions, right? So you might imagine that the DLPFC might have an inhibitory role over ventral striatum, but the relationship, of course, could go the other way. And so we subjected this to a formal mediation analysis. And what we found is that the ventral striatum was a full mediator of the relationship between the DLPFC and craving. And while we can't really determine causality from imaging data, which is correlational in this particular way, this does suggest a regulatory role for the dorsolateral prefrontal cortex over the ventral striatum, and a prefrontal striatal pathway that underlies the regulation of craving, especially using cognitive strategies in this particular context. Now, I want to highlight these two regions in DLPFC here in these data from this 2010 paper, because we've now replicated this across several different studies with a few different populations. These data were collected as part of one of Mark Batenza's grants. This is with cocaine users. Again, later over now, you see basically the same region being recruited. This is heavy cocaine users, right? So not cigarette smokers, really heavy cocaine users. Some people certainly would have predicted this wouldn't be the case. We see the same thing in the CTNA pilot alcohol data. Down here, in collaboration with Godfrey Perlson, we've now collected data from, I think, 70-something pre-bariatric surgery, highly obese individuals. We see, again, the same DLPFC region being recruited and later during the regulation of craving. And also, this is a really neat analysis that was carried out by Michael Stevens and Godfrey Perlson and their group. This is a whole brain regression predicting total weight loss a year later in that group that produced these data. And what you see is that those individuals who, before their bariatric surgery, recruit this region more strongly actually have the greatest weight loss a year later. I think this is really quite incredible. The last thing that I'm going to point out here is also a few years ago, we conducted a meta-analysis of all of the studies of emotion regulation, of negative emotion regulation in healthy adults. And what we see here, this is across 48 studies with healthy adults regulating negative emotion, and we see again the same region in the DLPFC being responsible for this kind of general emotion regulation, which suggests that again, under laboratory conditions, individuals are able to recruit this region and regulate their craving using somewhat similar machinery that healthy adults use when they regulate other kinds of emotions. So what do we talk about so far? So craving predicts outcome. It's a key process in addiction. We've identified some neural mechanisms that might underlie craving. And I've told you about CBT-based regulation of craving across these different kinds of stimuli. Oh, I didn't put alcohol in there. And specifically, I argued that the use of these strategies modulates neural activity in regions that are associated with craving, and that it depends on DLPFC. And I've shown you this now across drugs and emotion regulation in healthy adults. Now, the fact that there are meaningful individual differences, I think, has potentially the most clinical implications here. First of all, people have talked about this kind of pathway as a plausible mechanism for CBT-based treatments, which is something that actually we're currently testing with Brian Killick and with Kathy Carroll in a clinical trial for using CBT for cocaine dependence. We're actually using the ROC task as a probe both before and after treatment to see whether we can actually see this change with treatment. And also, it's suggests that we now have both psychological and neural targets to potentially develop, test, and improve current and future treatments. So already people have used ROIs from the papers that we've published on this to test TMS and TDCS, so brain stimulation in the DLPFC, which actually has been shown to reduce craving, and we're testing that now in the context of cognitive training. I will show you that momentarily. One reason, this is of course very exciting, but there's also reason for concern, and that is that the DLPFC is actually a region that has been often implicated in several abnormalities in heavy substance users. So this is a structural MRI from Arthur Brody from 2004 showing that healthy adults have thicker gray matter, greater cortical thickness in both right and left DLPFC. This is from cocaine users, Macros 2008, again, greater gray matter density in DLPFC. This is from Golnaz Tabibnia in 2011 with meth users. You can see this repeating itself with alcohol, and this is actually functional neural activity. This is from Hugh Garavan's group and Liam Nestor during a go-no-go, a cognitive control task, showing that healthy adults have greater activity in this region compared to cigarette smokers. And then we've shown, this is a paper with Elise DeVito and Mark Potenza and Kathy Carroll, during a Stroop cognitive control task that healthy adults show more activity in this region compared to cannabis users. So really we see a potential prefrontal abnormality, both structural and functional, across individuals with addiction, which suggests that if we rely on strategies that depend on DLPFC, we might be in trouble. This is also concerning because Amy Arntzen has shown very elegantly that stress severely impairs prefrontal cortex, and others have shown elegantly that stress literally leads to dendritic shrinkage in the prefrontal cortex. And of course, drug users certainly are very frequently stressed. And so this really led us on a search for strategies that might be less dependent on the DLPFC. We didn't know if we were going to be able to find them, but the kind of global question was, are all strategies created equal? Perhaps some of them might be a little different. And this is what brought us to the study of mindfulness. Now, while I don't have any official conflicts of interest, there is a little bit a kind of a disclosure that I need to make, which is I've been meditating for a long time, and I think that for me at least, mindfulness is a very powerful tool. I wouldn't say that I'm biased because I also think CBT is a wonderful tool, but I just thought I'd put it out there. This is me meditating on the beach in Costa Rica. Very different than being here. So what is mindfulness? Some of you might know, some of you might not. The classic definition is moment-to-moment, non-judgmental awareness of what is. And if that sounds a little foofy, there's actually an operational definition that divides mindfulness into two components: an attention component that specifically orients individuals to the present moment experience, right? So attention to what is happening to you right now. And the second component, which is the component of attitude. How are you relating to the present moment? And the recommended attitude is really one that's open, curious, and accepting of the moment exactly as it is. And the idea is that this really leads to a non-elaboration of present moment experience, even if it's negative, and a kind of a non-reactivity even to negative events. Or in the words of the Beatles, you just let it be. Now, mindfulness comes with a rich historical context, obviously. Since the days of the Buddha, it's a Buddhist practice. But importantly, it's actually part of a very large system, right? It's not a single thing. The goal is the cessation of suffering, or in more Western term, enduring happiness. And it's cultivated through mindfulness meditation. And importantly, it doesn't really need to be taught only in the context of a Buddhist approach. It's been imported into many, many Western treatments now: mindfulness-based stress reduction, mindfulness-based cognitive therapy, mindfulness-based relapse prevention, DBT, dialectical behavioral therapy, and acceptance and commitment. All are very well-established kind of studied treatments that have a mindfulness components to them. But just in case you haven't encountered this, I'm going to do another little demonstration. I'm going to ask you to close your eyes for a moment. And as you close your eyes, I'm going to ask you to just for a moment try to follow the physical sensation of the breath as it comes in and out of your body. First of all, just notice that your breath is moving spontaneously. You're not making it, you're not forcing it. It just moves in its own way. And you can also take a moment to maybe notice your body sitting on the chair, maybe your back, the weight of your body on your buttocks. You can maybe notice the sensation of your feet on the floor. And when you're ready, you can open your eyes. What I hope that you all notice is actually in a moment, it's very, very easy to experience mindfulness. What's really difficult is to string moments of mindfulness together, and that is the purpose of meditation practice. And research has shown that that kind of meditation practice that strings moments of mindfulness together is effective in reducing symptoms of stress and anxiety, chronic pain and pain intensity, and is also effective for treating particular disorders, including depression. There's a meta-analysis already on this, anxiety disorders, meta-analysis on this as well, social phobia specifically, borderline personality disorder, and also for altering biological markers that are associated with health, including increased telomerase activity, which is typically associated with youth and longevity, and reduced inflammation, pro-inflammatory gene expression, again, associated with health. And so some of the questions that we came into this line of work with is mindfulness effective as a treatment for addiction? Is it effective as a moment-to-moment strategy to regulate craving? And what might be the neural mechanisms? Are they really the same or different than CBT? I'm going to briefly talk to you about a randomized controlled trial we ran for smoking cessation. This was in collaboration with Judson Brewer when he was still here. We recruited treatment-seeking smokers and randomized them to receive either mindfulness training for smoking, which is a treatment that he and I adapted from mindfulness-based relapse prevention to be an active cessation treatment, and matched very well to freedom from smoking, which is a standard American Lung Association treatment for smoking cessation, which is a cognitive behavioral treatment. They're both active group treatments. They're matched for length. They were matched for a number of sessions. They were all administered over 8 sessions over 4 weeks. And just to give you a taste of how they're different, if freedom from smoking is CBT-based, mindfulness training is based on MBSR and MBRP, so really having a more of a mindfulness component. And if in freedom from smoking, there's emphasis on cognitive change, including thinking about, instead of craving, think about the negative consequences of smoking, behavioral modification and relapse prevention. In the mindfulness treatment, there's a lot of emphasis on attention and acceptance of negative emotion and craving. Quitting is set up as a goal. In FFS, it's set up as an aspiration in mindfulness treatment, again, with this kind of open attitude. FFS teaches cognitive regulation of stress and craving. Mindfulness training teaches mindfulness of stress and craving, not surprisingly. And then in lieu of behavioral strategies and weight management during smoking cessation, it teaches mindfulness meditation, body scan, and loving kindness, all kinds of meditations that individuals can engage in. In terms of the smoking cessation outcomes, I'm just going to show you the data for during the treatment period. What you see is that both treatments were effective in reducing smoking. When we looked at the follow-up data, mindfulness training was actually even had more sustained effects over time. But what we really wanted to know or to start to probe are the neural mechanisms that might underlie its effect on smoking cessation. This work was done in collaboration with Rajita Sinha. You probably all know her stress paradigm. Individuals come into the scanner. They receive instructions to imagine the story that they are about to hear. They actually produce this as a memory to a research assistant some weeks before who made it into a script, and then they listen to this scenario being described to them while they're being scanned. And so we looked at brain activity during the stress scenario after treatment between the freedom from smoking group and the mindfulness training group. And this paper is now in Press and Neuroimage in the Science of Change special issue that's coming out. So what you see here is that there's a few regions that are showing greater reactivity to stress in the freedom from smoking group compared to the mindfulness training group, specifically in the amygdala, in the putamen, the ventral striatum, and the thalamus, all regions that I also showed you before were associated with craving. And this is data extracted from the amygdala. We also see regions in the mid and posterior insula, and you see here also the freedom from smoking group is showing increased stress reactivity compared to the mindfulness group. What was really remarkable, however, is that across most of the work that was done on kind of cognitive regulation of emotion and stress is that we almost always see, I just showed you actually the meta-analysis, we almost always see dorsolateral prefrontal cortex. Here we see absolutely no more recruitment of DLPFC in the mindfulness training group, even though they're showing reduced reactivity to stress. And so, and I'm not going to elaborate on this too much, but keep this in mind because I'm going to return to this in just a moment. What we also did, meanwhile, is develop a version of the ROC task specifically with mindfulness instructions. So the stimuli were the same and the design was the same, but just the instructions were a little bit different. And the first study of these was done in collaboration with Cecilia Westbrook and Golnaz Tabibnia and David Cresswell at Carnegie Mellon. So the first instruction, just like before, focus on the immediate feelings. If you're shown a picture of a cigarette, you should think about how smoking will make you feel when you light up. Focus on the smell and how smoking the cigarette will taste. Here we use the keyword feel. Just feel your feelings. And the next instruction oriented participants to notice and accept without judgment, right? So this is the mindfulness instruction. And we specifically told them, if you see a cigarette, notice any thoughts, sensations, and cravings that arise. You can ask yourself, can I just be okay with this feeling? Simply notice and accept any sensations that you experience. And the keyword we used for this was the word accept. The trial time course looks exactly the same. They see an instruction. In this case, there's only cigarette stimuli, and then they rate their craving. So first, in terms of ratings, we see the same pattern that we saw before, when that acceptance is actually a very effective strategy for reducing craving in this population. These were all pre-treatment cigarette smokers. And what we see is also reduction in brain activity. This is a subgenual cingulate. It extends back into the ventral striatum during the accept instructions compared to feel. But what we don't see, again, is any recruitment of the dorsolateral prefrontal cortex. Not at all, actually. And this is where we started getting curious, right? We've now seen it across a few studies, and it really suggests one of two hypotheses. The first is that the PFC, the prefrontal cortex, is actually implicated in mindfulness-based regulation, but we just haven't been able to access it in this kind of data or in the way that we've probed it. And the alternative hypothesis is that mindfulness really might operate via different neural mechanisms compared to CBT and might not depend on the DLPFC in the same way that CBT does, or CBT-based strategies. And one way that we decided to test this was to actually use stimuli that were much better validated, and specifically negative emotion, where there's just hundreds of studies, in physical pain, because especially with physical pain, right, there's a particular temperature on people's arms, and we can really tightly control the intensity of the stimulus they receive. And so we can look perhaps better at the effective strategy. So in this study, we recruited adult healthy participants with no psychiatric or neurological disorders, and we showed them both images and applied temperatures to their arms. So we gave them neutral photos and negative photos. A neutral photo might be a picture of a chair. I'm going to show you something really gross in a second, so if you don't like it, don't look. We also showed them negative photos from the International Affective Picture System, like that right there. And then we also applied temperatures, including using a thermode on their arm that can be ramped to very, very specific temperature Celsius, warm temperatures that were kind of neutral, and then hot temperatures that were rather painful. And we calibrated this to people specifically so that we had, for each person, a warm temperature that they considered warm and not painful, and something that they considered the highest level of pain they were willing to tolerate, or an 8 on a 1 to 9 scale. And the instructions were very similar: react naturally to the stimuli or attend to the sensation without judgment. Just notice and accept any sensation that you experience in response to the aversive picture or painful heat. The trial structure, again, you've seen before. They get an instruction, they see images or receive heat with just a yellow fixation cross on the screen, and then their arm gets hot, and then they get to rate how negatively they feel. So first of all, just to make sure that you understand, the stimulus intensity effect was obviously quite strong. So people respond much more negatively to negative images than to neutral images, and to hot temperatures than to warm temperatures. And when we look within the negative images and the hot temperatures only for the effect of the accept instruction, people actually rate, and I'm going to focus on the temperatures here, people rate the temperatures as less painful when they're just noticing and accepting how they feel, right? Now remember, these two bars are the exact same Celsius temperature on their arm, but they're actually rating it as less painful. And when we look at their brain activity, we're actually seeing very similar data. I'm actually going to skip this slide with the images and just going to make the point on pain, because really, again, with temperatures, you can't lie. So here we see regions that are more responsive to hot than warm temperatures. So these are regions that are typically considered to be the pain matrix. They're responsive to hotter temperatures. And what you see is that very, very similar regions, when you look just within the hot temperatures, are actually less strongly recruited during the accept instruction, right? Same temperature, just different instruction, but we see less activity in sensory motor regions, right, that are considered to be direct representations of pain intensity, anterior insula, dorsal anterior cingulate, and the thalamus, all regions that overlap with the regions that are actually responsive to the intensity of temperature. I think that this is really compelling evidence. You can also see the extracted data here for where you see that the neural activity associated with the accept instruction on hot trials, this is in the insula and the thalamus, actually resembles much more the warm temperatures than the hot temperatures. And the only difference is that people are just noticing and accepting the sensations that they're experiencing. I think that that's really, really dramatic. We also ran this cool analysis with a neurological pain signature. I'm going to skip that as well. I see I was very ambitious today in how much data I was going to be able to show you. But I am going to point out that, again, we're seeing no increases in prefrontal cortex along with these reductions in pain intensity regions, right? Again, really, really striking data. And I think that now, with kind of these three studies coming together, we can really start saying that mindfulness-based regulation doesn't depend on prefrontal cortex, at least not in the same way that CBT-based regulation does. And what we're seeing really across these different studies is a reduction in reactivity rather than an increase in cognitive regulation when people are just noticing how they're feeling and kind of not arguing with it, letting it be exactly as it is. For those of you who know Buddhism, you might think about the parable of the second arrow. This is actually very consistent with what Buddhist psychology would predict would happen. I'm also not going to talk about, we also have some consistent data with experienced meditators. This is also work that I did with Justin Brewer and Patrick Warhunski some years ago, where we showed experienced meditators showing not increases in prefrontal cortex over time compared to novice meditators, but actually changes in resting state activity and connectivity. Okay, so I only have a few minutes. I'm going to talk about how we're translating this mechanism-focused research into testing and improving treatments, specifically through training. So craving is this basic process, right, with known neural targets that I showed you in the meta-analysis, and the regulation itself is also now a specific strategy-dependent target that we can look at. And so we started thinking about it as a new approach for maybe developing kind of a neurocognitive training, or a training that is brief and targeted in skill-specific based on the mechanisms that we already understand. And it's both theory-driven and data-driven. And I also should mention that in the context of anxiety disorders, this has been shown to be effective both as a standalone treatment and as an adjunct to other kinds of treatments. In our hands, we've basically had people regulate using the rock task just over and over in a manner of training, using often 75% of the trials as strategy trials, and we now have 5 studies. I'm only going to show you a tiny bit of data on this. Across all of these studies, we have typically a no training condition, a CBT, and at least one CBT-based training condition, and some of them also have now a mindfulness-based condition. And we also have varied the dose from 15 minutes to 6 hours of training in this across the different studies. And the first study that I'm going to show you really briefly is a food choice study, again with Becca Boswell. Participants make 108 choices between healthy and unhealthy foods. Then they go through a rock training. The main type that I think is easiest to explain is the one where they practice this negative strategy with unhealthy food. It looks exactly like the task itself looks. And then they again make food choices. This is all one session in our lab. We also did a parallel training condition helping them practice thinking about the positive consequences of healthy food. It's actually quite remarkable. People end up agreeing to pay more and want broccoli more. And also we have a control condition where they just look at images, so we're controlling for exposure, but not training them on anything. And what I'm going to show you here is a change in healthy choices from pre to post training. Remember, this is just 15 minutes of training. What you see, and this is, by the way, 420 participants in the first study, what you see in the look condition where they just get exposed is people actually make a little bit more unhealthy choices compared to healthy choices on the second choice task compared to the first when they get no training. But if they get either positive training or negative training, so think about how good healthy food is or how bad unhealthy food is, they show a significant increase in their choices of healthy compared to unhealthy food. These differences are significant, not between the trainings, but between both trainings and the look condition. When we first saw this, I swear to God, I didn't believe it. I thought that this could not be, and so I made Becca run the same study again on another really, really large sample. Thank you, Becca. And what we did was replicate those data exactly. So slight reduction in healthy food choices in the look condition, an increase in the positive condition, an increase in healthy choices in the negative condition, both significant. Of course, one might say that these are just food choices in the lab. We're now also running a study looking at actual eating behavior, or are starting next week to run a study looking at actual physical eating behavior after the same kind of training. But we've also, meanwhile, run a study with cigarette smoking. We're going to call these studies 3 and 4. In these studies, there's a lab session in the lab where they get either a no training control and then either 30 or 60 minutes of training on the CBT-based kind of negative strategy. And then we do a phone follow-up 1 week week, 2 weeks, or 1 month later, looking at their smoking. And what you see here, and we're literally currently writing up these data, is that the group that got the training happened to be randomized into the training actually started smoking a little bit more, but was smoking less 1 month after treatment, and certainly a week after treatment and right after training. This is actually daily diary data, the pre and the post. This is phone follow-up. And this is a 37% reduction in cigarettes per day, which is quite amazing for 30 and 60 minutes of training. And when we looked at whether the training ratings themselves can predict outcome, what we see is that their ratings during the training itself, the lower craving they got with the strategy, the less they smoked at follow-up, which I think is also quite extraordinary. We're currently running a much larger version of this as a stage 1 RCT of ROC training. This is project 3 in the Psychotherapy Development Center, of which I'm PI. It includes 6 lab sessions that are an hour each, a follow-up up to 4 months. We do the fMRI version of the ROC task with both mindfulness and CBT-based strategies, but then they really are randomized to receive only one of these treatments. And then, the training looks exactly as before. I want to leave a couple minutes for questions, so I'm going to totally not tell you about the behavioral data, although this is the pilot data. This is not from the ongoing clinical trial. This is the pilot that we ran. This is the what served as our pilot data for the grant itself. And what we're seeing here is that the mindfulness-based training is actually associated with general decrease in craving, whereas the CBT-based training is only associated with decrease in craving during the strategy condition itself. What we're also seeing really quickly is that when we look at brain activity during the CBT-based training, we see more recruitment of DLPFC during regulation after treatment compared to before, but we're seeing absolutely no changes in DLPFC in the mindfulness-based training. All right. That was a fun ride. Addictions are severe conditions with tremendous costs to individuals and society. There's really an urgent need for better treatments, especially given the fact that those are the most common psychiatric conditions. Craving is a key factor in addiction. I hope that I convince you that it predicts drug use and therefore is a good target for study. It's implemented in regions that are associated with dopamine, with emotion, with the default mode, and with motivation, like the ventral striatum and the amygdala. While regulation of craving, which is clinically important to counteract craving, is also a core feature of treatments like CBT and mindfulness-based treatments. Strategies that regulate craving also regulate activity in these brain regions. And then I made a really important point that I hope that you'll take with you after this talk. CBT and mindfulness-based treatments may seriously rely on different neural mechanisms, and specifically mindfulness-based treatment may reduce reactivity without depending on recruitment of prefrontal cortex, really raising the argument for bottom-up versus top-down kinds of treatments or kinds of regulation. Some of the places where we're taking this is obviously investigating ROC training as a mechanism based for treatment of development, but also for testing of treatment, which I mentioned before. We've already shown that it improves food choices, that it reduced smoking compared to no training, and we are currently running our randomized controlled trial comparing these two. I also just received a really good score on this R1, so if you like me, you should pray for me that this will actually get funded. This grant will promise to test the boundary conditions of the effects of stress, specifically on cognitive-based regulation, so that we may better understand what happens to drug users when they fail to regulate in moments of stress, and I hope that you will all stay tuned for that kind of work. And before I end, I'm going to come back to craving. I mentioned to you that the Buddha said that craving is the cause of all suffering, that Spinoza said that desire is the very essence of man, but it was really John Locke who kind of predicted this line of work when he said the discipline of desire is the background of character. Thank you all for listening.